Welcome to Heightened Characterization of Therapeutic mAbs using a novel enzyme-facilitated middle-down approach. I'm Tamlin Oliver, Managing Editor of Genetic Engineering and Biotechnology News, and I'll be the moderator for this webinar. Whether you work in the development or production of monoclonal antibody therapeutics, this webinar will be of interest to you. I'm not telling you anything you don't already know when I say that quality control and analysis of mAbs is essential throughout therapeutic development to production. What you might not know, and what this webinar is focused on, is the Antibody Subunit Domain Mapping Assay, a new characterization method that is enabled using ultra-high-resolution mass spectrometry and a novel IgG-specific enzyme. Our two speakers today, Rob Duffield, Senior Principal Scientist at Pfizer, and Frederick Olson, Chief Operating Officer at Genovis, will talk about the use of the FabRICATOR enzyme and how it facilitates the characterization of multiple post-translational modifications. They will also explain how these novel enzymes can quickly generate specific antibody fragments, how this enzyme can be used for rapid comparability studies of biosimilars versus innovator compounds, and how these enzyme systems can be used to screen production clones, among other benefits. Rob is up first, and he will talk about his use of antibody subunit domain mapping. But before he begins, I want to encourage you to submit questions for our Q&A session at the end of the presentations. We will answer as many questions as we can. Simply type your question into the Ask a Question box and hit Submit. Rob, let's get started.
Thanks, Tamlin. Good afternoon. Today, I'd like to speak to you on heightened characterization of therapeutic mAbs. Today we'll be illustrating how we use a novel enzyme facilitated and enables us to address a middle-down approach, a characterization strategy for mammalian-derived protein therapeutics. For the outline today, we'll be going at a high level, giving a general heightened characterization strategy. We'll be moving into the antibody structure and characterization targets, followed by getting into the heart of the technology that we'll be utilizing to establish this middle-down characterization approach, and that is the Bruker Daltonics Maxis ETD ultra-high-resolution QTOF mass spectrometer. In addition, it's utilizing the FabRICATOR enzyme offered by Genovis, which is a novel IgG-specific enzyme. Next, we'll be showing a couple applications where we utilize the enzyme and the technology to create an antibody subunit domain mapping strategy. First, we'll be looking at antibody clone selection to establish sequence fidelity. Next, we'll be looking at an offline evaluation of linker integrity for antibody drug conjugates, and also a general characterization of other antibody drug conjugates, followed by a summary to wrap things up. So the characterization approach for antibody therapeutics can be kind of categorized in three segments: a top-down a top-down proteomics approach, a middle-down, and a bottom-up. The bottom-up and a top-down strategies is a well-known proteomics strategy where the bottom-down really focuses on taking your product and whittling it down to its counterparts of the proteolytic digest and doing a local post-translational modification ID, working on quality separations, relying on accurate mass, and then using a couple other novel gas phase fragmentations, CID or ETD. A middle down focuses on the top down focuses on the intact protein to go after accurate mass, as well as a more global post-translational modification ID for the protein. For middle down, here we will utilize the FabRICATOR enzyme to do a specific digestion of the mAb and get a regional post-translational ID for the various modifications. We will rely on the mass spectrometry for the accurate mass, as well as UPLC for quality separation. Here we have the antibody characterization targets for a general therapeutic mAb. Depicted is an IgG2. The major characterization targets are the post-translational modification in glycans, as well as the disulfide bonds, and then looking for the general sequence fidelity of the mAb. There are several other attributes that could be accounted for, including oxidation, deamidation, C-terminal lysine, and a few other modifications that would be of interest. To get at some of these modifications and to establish the sequence fidelity of the mAb, we utilize the Bruker-Daltonics Maxis 4G ETD ultra-high-resolution QTOF mass spectrometer. This particular instrumentation is able to deliver mass accuracy up to 600 parts per million. The mass resolution is phenomenal. We have 60,000 resolution. That's a full scan resolution and a superior scan speed at 30 spectra per second. This particular instrumentation is utilized for a number of applications, including biomarker discovery, trace analysis studies, small molecule. It's the bottom-down approach for protein therapeutics as well as top-down. But today, we'll be utilizing it for antibody subunit domain mapping, which is a middle-down approach. Before we get to using that technology, we're introducing now the FabRICATOR enzyme offered by Genovis. This particular enzyme is also referred to as IdeS. It's an IgG degrading enzyme, and it's derived from streptococcal pyogenes. It's a very novel cysteine protease that it cleaves the gamma chains of human IgG rather specifically. At this point in time, there's no other known side reactions utilizing this enzyme. It has a very specific cleavage site at the heavy chain, right below the glycine or alanine of IgG 1, 2, 3, 4 isotypes at 235, and it essentially liberates a Fab dimer and 2 single-chain Fc domains. Using this enzyme, we're able to capture these domains and maintain them at a neutral pH in a really brief time and minimize any side reactions that could occur when handling your sample. The next step that we would utilize after the first digestion using the FabRICATOR would be through a mild reduction. With that reduction, we're able to take the Fab dimer and reduce it down to both the FD prime domains and the light chain subunit. We also will still have the single chain Fcs, but now they would be completely reduced. So now you can see that we have 3 primary components of the mAb. We have the light chain subunit, the FD domain, as well as the single chain Fc domain. Now the single chain Fc domain will enable you to get at the glycosylation pattern, whereas the light chain subunit will enable you to get at any light chain CDR region for modifications or characterization. And the FC or the FD domain would enable you to get at potential modifications in the FD region or the hinge region if it's not including a disulfide bond. Here we show this particular approach, taking the FabRICATOR down on a mAb and then doing a complete reduction, and now we have the separation. This separation shows the power of the technology altogether, where we have the use of the enzyme, the mass spec, and now the separation on the UPLC. We have the separation of the single chain Fc, which is the first peak shown in the chromatogram. We have the second peak shown, the light chain, and the FD. On the bottom of the slide, what we have now is a comparison of the observed monoisotopic masses for each of these species compared to the theoretical, which is simulated based on the known amino acid structure of each of these domains or subunits. You can see the great mass accuracy that's obtained utilizing the Maxis mass spectrometer for each of these species. They're all less than 1 part per million, with the exception of the FD, which is just a little bit over 1 part per million. What this enables us is to get at the sequence fidelity of each of these domains and subunits for a given monoclonal antibody. We use this technology to establish sequence fidelity in a general scheme when we nominate a clone. Here we have a slide depicting a various number of analytics for product quality assessment. Within the industry, there are a number of attributes that we look at. One is the high molecular weight species, or dimer, or aggregate, if you will. Another is the in-link glycosylation pattern for a given mAb, and another is charge heterogeneity. These three techniques, or these three attributes, are followed throughout the development for monoclonal antibody. But before we can get to that, we need to ensure that we have sequence fidelity, or to ensure that the clone that we select is generating the appropriate mAb with the appropriate sequence, or the expected sequence. We utilize the subunit domain mapping assay to do just that, to ensure that we have sequence fidelity. There are other approaches that are used concurrently, and that is offline peptide mapping, as well as some ctDNA technology, PCR, to get at the sequence fidelity. Here what's shown is a general schematic view of a manufacturing process where you go through all the bioreactors, the purification columns, the UFDF, leading to your purified mAb. But before we can get to that, we need to develop the clone. In selecting the clone, we use both the Maxis and the Maxis mass spectrometer and the FabRICATOR enzyme. We use this to ensure that we have sequence fidelity. Here we show the traditional antibody subunit mapping without using a FabRICATOR or the Maxis mass spectrometer on the heavy chain. You can see that on the bottom trace that for the light chain, we do get monoisotopic resolution for the light chain. However, for just the heavy chain, using high-resolution mass spectrometer, we're not able to get at the monoisotopic resolution or have a confident mass spectrum for the given heavy chain. Therefore, we could potentially miss the modifications. The slide here does show the known modifications for this given heavy chain. However, without using a FabRICATOR approach, we would not be able to get to this particular detail. When we take that same molecule and go through the antibody subunit domain mapping or the clone selection process, you can see that we'll be able to get at the detail that's necessary. Here we show a clone A versus a clone B. In clone B, we have a great separation of both the light chain subunit and the two domains of the heavy chain, the single chain FC and the FD. For the particular clone called clone A, what we see is an array of additional peaks. They are associated with the single chain FC as well as the FD. We will focus primarily on the single chain FC modification, which we refer to as a threonine 304 proline modification. That particular mass spectrum for the single chain FC for the native and the modified is depicted here. What you see with the theoretical mass is low parts per million mass accuracy, but compared to the modified form, you can see that we have a less than 3 Dalton mass shift. Using the high-resolution mass spectrometry, we're able to narrow down the very accurate mass determination of what that modification could be. And this is the technology we use to deduce the structure at a regional level. So a threonine 304 proline modification. This is an example where sequence fidelity is important, as well as having the right tools to utilize to get to these modifications. There are other examples or applications of using the enzyme FabRICATOR. You can utilize it for an array of mAbs, IgG 1s, 2s, 3s, and 4s, bispecifics, and other numbers of antibodies. You can also utilize it for COVAX bodies, which is a type of fusion antibody. There's also antibody-drug conjugates, nanobodies, and N and C-terminal FC fusions. We will focus here on the use of the enzyme with COVAX bodies. The first example will be used for COVAX body 1. Here, this particular COVAX body has a reactive lysine. It'll be lysine 99 on the heavy chain of the antibody scaffold. The lysine residue is rather reactive. The epsilon amino acid of the lysine 99 is reactive to various components of a linker. Here we have a TSP1 mimetic that is conjugated to the antibody to create a ADC with a higher mass of about 152,000 Daltons. Using the subunit domain mapping analysis approach, we're able to deduce the integrity of the conjugate all the way down to the FD prime. Looking at the single chain FC, you can see that we have minor modifications that includes oxidation and some clipping that's observed, but notice there's no conjugation occurring in the single chain FC. Same thing for the light chain. The light chain integrity has been confirmed. And then moving to the FD component, which is the upper part of the heavy chain, you can see that you have the conjugation reaction occurring there. You can use this technology offline to evaluate the level of unconjugated FD that is occurring or not occurring. Whether offline or online, you can still go at the fidelity with mass spectrometry. Using the high-resolution mass spec Bruker Maxis QTOF, we're able to confirm that the single-chain FC has the integrity as anticipated, as well as the light chain, the integrity remains. Ultra-high resolution is also applied to the FD. Here we show the theoretical mass for the FD component, which is the unconjugated region, and it agrees quite nicely with the theoretical mass. Same thing for the conjugated FD. So using this technology, you're able to confirm the conjugation integrity using high-resolution mass spec and the novel enzyme FabRICATOR. Now moving along to a more exciting approach is using the enzyme to get at more of the integrity of the linker for this particular COVEX body. Here we have a COVEX body 2. This particular COVEX body is a bispecific where we have a VEGF binding peptide as well as a Ang2 binding peptide. Within this, they're connected through a rather complex linker that involves the use of a maleimide chemistry. Within that chemistry, we have a succinimide ring, which we'll be focusing on to ensure that we have the integrity for this particular linker. For ADCs, maleimide chemistry is used quite commonly. What we're focusing on is the open ring formation that could happen once the ring is hydrolyzed. Hydrolysis of the substituted succinimide ring leads to a mass increase of a little over 18 Daltons. It also increases the overall acidic charge for the given protein. These are both quality attributes that would have to be monitored and controlled throughout both manufacturing and storage of the biotherapeutic. Doing just that, we developed an assay to monitor the open ring structure and the closed ring structure. Now, to repeat, the closed ring structure is the native version. The open ring is a degradation product, so that forms a acidic charge. It's a hydrophilic modification that we're now able to monitor utilizing a similar separation using UPLC. The separation below depicts the single-chain FC and various related components of the single-chain FC. We also have the light chain and a few light chain-related components. But looking to the far right of the chromatogram, we have the hydrolysis event that occurs just at the linker. So we have the closed form as well as the open form. This is monitored throughout stability, as well as through the manufacturing process for this particular ADC. This depicts another example where the enzyme is quite powerful to get at the integrity of a linker or the integrity of our antibody drug conjugate. To finish up, I'd like to summarize the use of this enzyme where we provided heightened characterization of therapeutic mAbs is enabled using this novel enzyme. FabRICATOR is an excellent and very specific protease that facilitates mAb digestion, and not just mAb, but mAb-likes, so ADCs, FC fusions, COVEX bodies, and a number of other mammalian-derived modalities. This approach, the antibody subunit domain mapping, allows confirmation of antibody sequence, so sequence fidelity, which is shown for our clone selection. It also enables us to go after the activated payload structure for a given ADC, and it provides rapid information about regional post-translational modifications like glycosylation, the C-terminal lysine, methionine oxidation, and deamidation, which is also included in storage-induced modifications. This technology allows us to go quickly after the heightened characterization for a number of protein therapeutics that are mammalian-derived. For acknowledgments, I'd like to thank a number of colleagues, particularly our leader, Marta Ciprin. She is the VP of Analytical R&D here at Pfizer. We also have Thomas Porter and Jason Rouse has supported the technology development for the analytics and applied towards mammalian-derived protein therapeutics as well as vaccines and other protein therapeutics. There's a number of colleagues in the mass spectrometry group that has led and continues on developing this assay further. James Carroll, Keith Johnson, Heather, Matt Thompson, and we also have a number of analytical scientists that have helped out tremendously, Jeff Reisig and Jason Starkey. Of particular note, I'd like to thank Libby Yates for her efforts in the initial proof of concept using this enzyme in 2008. And with that, I'd like to thank Genovis AB, Jen, as well as COVEX folks, Bob Murphy and Ryan Peterson. At this particular time, I'd like to hand it over to our next speaker.
Thanks, Rob. Before we move on to our next speaker, I just want to tell you that you can submit questions for Rob now by typing it into the Ask a Question box and hitting Submit. Frederick, we're ready for you now.
Thanks, Emily. So the outline of my talk will start off by covering the Fabricator enzyme, its background, the high specificity for IgGs, best reaction conditions, and different product configurations of the enzyme. I will then describe the endoglycosidase IgG0, which is recombinantly modified NOS, and how this is used in an optimal way. I will also show different product configuration using this enzyme. Later, I will highlight some different applications using either of these enzymes or both in different ways in terms of characterization of biopharmaceutical monoclonal antibody-based products. And finally, I will say a few words about our pipeline of new enzyme and how you can participate in the beta testing program of those particular enzymes. Both of our current products on the market, the Fabricator and the IgG0, as well as the enzyme in our product pipeline, has one thing in common: the ability to modify antibodies in precise and specific ways. And from an analytical point of view, this is, of course, of importance as monoclonal antibody-based product is complex to analyze as this. Fabricator is Genovis' trade name of IdeS, and IdeS is an acronym for immunoglobulin degrading enzyme from Streptococcus pyogenes, which is a human pathogen that can cause severe sepsis upon infection. IdeS was discovered by Professor Lars Björk at the Department of Infectious Medicine at Lund University back in 2001. The biological role of this enzyme is to circumvent the host immune response, and it does this in part by excreting the IdeS enzyme, which is then cleaves the host IgG pool into Fab2 fragments and FC fragments. Fabricator is a cysteine protease belonging to the same family as Papain. However, it is much different from other known cysteine proteases in that it's highly specific for IgGs. And to date, there are no other known substrates besides IgG for this enzyme. Genovis introduced a recombinantly modified version of this enzyme back in 2007. And Genovis also holds the exclusive license for use in biotech applications. The very high specificity of the Fabricator enzyme is to some extent correlated to the amino acid sequence of the substrate. This slide shows the Hinge region and the beginning of the CH2 domain of the human, mouse, and rabbit IgG. Fabricator cleaves all subclasses of human IgG as noted on the right side of each sequence. The cleavage is located between the two glycines, as indicated by the arrow on the top right. The exception is human IgG2, where cleavage takes place between the alanine and glycine. The rate of cleavage is much slower on IgG2, which indicates the difference in cleavage susceptibility between the LGGP motif found in the other human subclasses and the VAGP motif found in IgG2s. Looking at the mouse IgGs, there's a significant difference between the IgG1 and the other subclasses in the potential cleavage region. Indeed, mouse IgG1 is not cleaved even when incubated for extended time periods or with a very high enzyme to substrate ratio. The two mouse subclasses that are amenable to cleavage, namely IgG2A and IgG3. Although these two subclasses have the identical 4 amino acid motifs as human IgG1, the rate of cleavage is slower, which indicates that correct amino acid sequence itself is not sufficient for optimal cleavage. Indeed, it has been shown that synthetic peptides that contains parts of the amino acid sequence covering the cleavage site is not cleaved, indicating that the three-dimensional structure also contributes to the substrate specificity. The high specificity of the Fabricator enzyme have some interesting advantages in terms of the reaction itself. First, compared to other commonly used proteases like Papain or pepsin, there is no risk of overdigestion. Adding the fact that the enzyme is very rapid, this means that there is no need for doing optimization of reaction condition before applying the protease in real situations. Highest activity of the enzyme is achieved at 37 degrees, but the reaction can be performed at lower temperatures if incubation time is prolonged. For instance, at room temperature, we usually advise a 90 minutes to 2 hours incubation time for complete cleavage. Optimal reaction pH has been determined to 6.6, but from a practical point of view, there's little difference between applying the enzyme between, let's say, pH 6 to 8. And unlike Papain, FabRICATOR does not need the addition of cofactors like cysteine to have catalytic activity. The enzyme can be directly added to the solution containing the antibodies. So basically, it works right out of the box. Taken together, these characteristics of the enzyme allows for short incubation times at neutral pH, which minimizes the risk of sample preparation-induced modification of the target, something commonly observed using triplet digestion. So the basics of using the enzyme is quite straightforward. The lyophilized enzyme is reconstituted using water. Then the required amount of units is added to the antibody, and the reaction is allowed to proceed at 37 degrees for 30 minutes. And at that point, basically all material is digested into Fab2 fragments and intact FC fragments. The Fabricator enzyme is flexible regarding choice of buffers. Many commonly used buffers are compatible without any significant drop in activity. This table shows some of the buffers we have tested in-house that works. However, it is likely that other buffers not listed here will work equally well. Also, again, there are no need for cofactors or reducing agents to be added for enzyme activity. The general workflow when using the Fabricator enzyme in liquid chromatography mass spectrometry applications starts with a 30-minute digestion of the antibody at 37 degrees. After digestion, the antibody is further reduced and denatured to obtain individual single-chain FC, light chain, and FT fragments. This requires reduction using either DTT or TCP. Most published protocols use DTT or TCP in concentrations between 50 and 100 millimolar. Somewhat lower concentrations can be used with TCP. The reduction step is usually done simultaneously with the denaturing fragments using either guanidine hydrochloride or urea in concentrations of 4 to 6 molar. The sample preparation time is commonly between 60 to 90 minutes, and samples can be directly analyzed by LC-MS. The total time for sample preparation and analysis is only a couple of hours, which is time-saving and potentially allows for high-throughput analysis and automated workflows. The Fabricator enzyme is available in several configurations. It's available as lyophilized powders in different amounts, ranging from small amounts, individually packed for just adding the enzyme and run, to higher amounts for aliquoting or digestion of larger amounts of IgG. It's also available as a customized format upon request. Along with the lyophilized enzyme, we have also developed immobilized enzyme in spin column configurations. The immobilized enzyme is available for digestion of a few hundred micrograms up to 100 milligrams, as well as kits that allows for subsequent isolation of fragments. The kit is used for fast preparation and isolation of Fab2s and FC fragments. It consists of one column with the FabRICATOR enzyme immobilized on agarose and a second column containing Fc-specific capture select resin from back in Netherlands. First, the antibody is fragmented for 30 minutes on the fragment column, and then the content is added to the capture select column for removal of Fc fragments. Later, the Fc fragments can be collected using a standard low pH elution. This kit has become interesting, particularly in the preparation of antibody fragments for crystallization experiments. The key aspect here is the homogeneity of the fragments generated by the FabRICATOR enzyme, as this is of particular importance when growing crystals. Next, I'm going to say a few words about the IgG0 enzyme. The source of the endoglycosidase EndoS, with trade name IgG0, is also from Streptococcus pyogenes. The biological role, again, is host defense, as the enzyme deglycosylates the N-glycans associated with the Fc part of IgGs. This property is unique, as other N-glycosylated protein is unaffected by IgG0. So even in complex environments like blood or growth media, only N-glycans from the Fc part of IgG is hydrolyzed. This is another example of a very high specific enzyme from Streptococcus pyogenes. The enzyme was discovered by Professor Matthias Collin at Lund University and first published in 2001. The N-glycan is cleaved off right after the first glucanac, leaving one glucanac and, if present, the fucose, still attached to the peptide. The enzyme removes essentially all N-glycans except some high nanostructures and bisected glycans. Even in the presence of other N-glycosylated proteins, for example in serum or growth media, only N-glycans of Fc fragments are hydrolyzed. If present, N-glycans on the Fab part of an IgG is not hydrolyzed. There is no need for denaturing the IgG before the enzymatic digestion, and enzymes can be added directly to solution for digestion. Reaction time is 30 minutes at neutral pH and 37 degrees. It is possible to do co-digestion with FabRICATOR simultaneously as the Fc deglycosylation. Both enzymes can be added to the solution containing the antibodies and incubated for 30 minutes, yielding deglycosylated Fc and Fab2 fragments. Now I'm going to show a few applications of the enzyme in relation to characterization of antibodies. In this example from Daiwei Chen and Kevin Ray at Sigma Aldrich in St. Louis, they outline a workflow combining FabRICATOR, IgG0, and P-enjase-F for determining both Fc glycans and Fab glycans on a monoclonal antibody. Different glycoforms on both single-chain Fc and FD domains can be mapped using this workflow. Using high-resolution mass spectrometry, glycans present at approximately 1% is usually clearly detected. The size reduction accomplished by the use of the FabRICATOR enzyme, together with the site-specific deglycosylation by IgG0 and the more general deglycosylation using P-enjase-F, allow for glycan mapping of different sites in the IgG. In a recent paper by Michael Boyne and co-worker at CEDAR FDA, a U.S.-approved monoclonal antibody was compared to a biosimilar version from India using the middle-down approach with FabRICATOR digestion and subsequent reduction. As shown on the right, they could clearly and rapidly identify significant differences in the intensities of the observed glycan forms between the U.S.-approved rituximab and the biosimilar version. Other post-translational modification associated with the light chain, single-chain Fc, and the FD fragment could also be distinguished between the two. So in summary, it proved suitable using this straightforward middle-down LCMS approach for the identification of structural differences between the drug products from two different international sources. Another interesting application that has taken advantage of the enzyme's strict specificity for antibodies is described by Andrew Götz and co-workers at Amgen. In this case, endogenous IgG from patient samples was first deglycosylated in serum by IgG0, followed by the addition of the FabRICATOR enzyme, also in serum, for digestion of IgGs. Initially, they used a protein A purification step to isolate the Fc before doing LC-MS, and the corresponding chromatogram is shown in panel B. However, they later discovered that they could run the digested and deglycosylated sample directly on LC-MS without prior purification of single-chain Fcs and still readily detect the degree of fucosylation for both IgG1 and IgG2, and this is shown in panel C. Continuing on the same work, the results obtained by the fast preparation using the combination of FabRICATOR IgG0 together with LC-MS was compared to peptide mapping of tryptic digest for identification of glycopeptides. The figure shows good agreement between the FabRICATOR IgG0 combination method and the tryptic peptide mapping method in determining the degree of fucosylation. This paper is particularly interesting as digestion and deglycosylation of IgGs takes place in such complex environment, showing the very high specificity of these enzymes. Using FabRICATOR enzyme, it's also possible to create Fab prime fragments using 2-mercaptoethylamine. This is a protocol adapted from Hermanson's book Bioconjugate Techniques. This mild reducing agent is selectively reducing the tiles in the hinge region, thereby creating Fab prime fragments. This is especially interesting for bioconjugation, as the reduced tiles is readily amenable for conjugation using maleimide chemistry to various surfaces, nanoparticles, or other types of solid support. Conjugation to the hinge tiles is also oriented in a way that fragments is conjugated in an optimal position for binding to the antigen. And a few words about our upcoming enzymes. Currently, we have 2 additional enzymes in our pipeline that are approaching market introduction. The first enzyme on the left is a cysteine protease with a different cleavage site in the hinge region than the FabRICATOR enzyme. We are currently investigating this enzyme on various mAbs and establishing user protocols. The second new enzyme we plan to introduce this year has a working name of EndoX, and just like EndoS, it deglycosylates the end-linked glycans from antibody Fc fragments, but it appears to have activity also on more complex end-linked glycans commonly observed on recombinant IgGs, especially high mannose and bisected glycans. And for those of you who are interested in beta testing of these enzymes, you are welcome to contact me, and my email is at the bottom of this particular slide. So in summary, I've described 2 highly IgG-specific enzymes from Streptococcus pyogenes and how they can be applied in characterization of antibody-based biopharmaceuticals. Both enzymes are robust and rapid, requiring little optimization and little hands-on time. The very mild conditions and short reaction time also minimizes the sample prep-induced modifications. And finally, the enzymes can be applied at various monoclonal antibody-based products at different stages in development, production, and quality assurance. I would like to thank the group at Pfizer, especially Rob Duffield, James Carroll, Jason Rouse, Lisa Marcelli, Paul Brown, and Matthew Thompson for their work using the enzyme. At Sigma-Aldrich, I would like to thank Jiawei Chen and Kevin Ray for their work on the glycan mapping on different map sites. At Bruker Daltonic, I would like to thank Wolfgang Japs for his work in our collaboration on automated sample prep and workflow using the FabRICATOR enzyme. I also would like to thank Martin Hedström at Lund University for his work on the enzyme contributing to the products we have today. And finally, Tamlin Oliver from Genetic Engineering News for hosting this webinar. Thank you.
And thank you, Frederick, for the great presentation. Before we start the Q&A session, I just want to remind you one final time, this is the time to submit questions for either Rob or Frederick. Please just type a question into the Ask a Question box and hit Submit. I also want to let you know that we will be deploying a post-webinar survey. Please take a few minutes and respond. Your comments are extremely valuable to us. Okay, we're ready now. We have a lot of good questions. We'll answer as many as we can. Okay, our first question is for you, Rob. Will you comment on the LC conditions used for the mAb fragment separation?
Absolutely. For the LC conditions, while we can't go into the actual specifics, the separation was performed using a Waters H-class UPLC, and the column was a UPLC C4 column, and it was operated at an elevated column temperature. The mobile phases are rather quite common. They include acetonitrile, water, TFA, and IPA. The separation uses three different lines to, generate the separation that you saw. Thanks, Tamlin.
Okay, so the next question, Rob, for you is, for sequence variant analysis for clone selection, what is the quantization limit using the subunit domain mapping assay?
Using the assay that we showed the preliminary sensitivity is below 1%. However, we have not formally established this as the LQ. That's something that we will be working towards. At the moment, right around 1% low.
Okay, can you comment on the preparation to analysis time required to perform the antibody subunit domain mapping assay for the analysis of sequence variants?
Sure. Yes, the enzyme is quite robust, and it's very efficient. So the FabRICATOR enzyme allows us to expedite the digestion procedure, and it's not typical like trypsin or Lys-C where you'd have a 4-hour or overnight digest. The overall time for starting with purified or protein A purified mAb, we're looking at about 3 hours from the purified mAb to having ultra-high-resolution MS results in hand. Thanks. Okay.
Rob, have you tried to combine use of the FabRICATOR and IgG0 to enable the heightened characterization of mAbs?
Yes, this is another good example or a question where the example that we showed in this presentation of the clone A and clone B, we took an approach to use the FabRICATOR and the IgG0, but just in the opposite order. First, we removed the N-glycan, whittled it down to the core glycnac with or without the fucose, followed by the FabRICATOR digest. And the goal was to reduce the heterogeneity in the single-chain FC that's associated to the N-glycan. That approach allowed us to go in and do more of a gas phase characterization using electron transfer dissociation, using it on the Bruker axis, ultra-high-resolution mass spec. Thanks.
And this is a perfect segue to the next question. Someone asks, how is a Maxis MS calibrated using a small molecule calibrant or a large biopolymer of known mass?
Yes, I mean, there's a number of calibrants that can be used. Any of the mass spectrometers out there probably would have their own preference. We utilize the Agilent high mass calibrant, which is a small molecule calibrant.
All right, Frederick, the next couple of questions are for you. How specific and consistent is the FabRICATOR enzyme?
Well, it's the only known substrate today is actually IgGs and human IgGs and some of the other IgGs like rabbit and some of the mouse then, as I mentioned. And the specificity extremely high, so even if you have a very complex pool of glycosylated or other types of serum proteins or growth media, if you throw the enzyme in, it will only cleave the IgGs.
All right, so the next question someone asked is, can it be used for rodent IgGs?
Yeah, we know that, for example, IgG2A and IgG3 of mouse works. We haven't really tested, let's say, rat, and I'm not aware of anyone who has done that successfully either.
All right. Regarding high throughput, do you have any new product formats coming out to make it even more straightforward?
Yeah. We do actually working together with Bruker on a more of an automatic workflow where we have the reducing agent and the denaturing agent also in a second tube lyophilized for rapid sample prep and convenient sample prep. which we are evaluating. And I think we will have a poster on this year's ASMS on that approach.
Okay, and what are the benefits of using FabRICATOR enzyme compared to Papain or limited Lys-C digestion for a middle-down approach?
Well, I will have to say it's, again, the single-site cleavage compared to those enzymes because you might end up with having actually multiple cleavage sites. And also, there's really no need for optimization of the reaction conditions. So from that point of view, it's very fast and it's easy to handle.
All right, and can the immobilized FabRICATOR resin be used in larger scale setup, for example, gram scale, let's say a few hundred grams even?
Yeah, sure. We have actually done some work on that in-house as well, where we have used the immobilized enzyme to fragment large amount in an ECTA type of setup in an ECTA system, where we just pump the antibodies through the column and receive the fragmented antibodies, and then we can do purification of the individual fragments.
Okay, so we have a listener who apologizes first in case they missed this if you already said this, but they wanted to know, what is the stationary phase of capture select?
It's agarose, and the resin is from back in Netherlands, so it's a llama antibody fragment that has been raised against the conserved region in the FC of IgGs, which is immobilized onto agarose.
Right, and can you give an example or name of a protein that was characterized using FabRICATOR other than the mAb?
Yeah, sure. We know that it has been used, for example, for Embrel type of FC fusion proteins in characterizing glycan or mapping glycan sites on FC fusion proteins, for example, yes.
Okay, another question for you, Frederick. Typically, what is the enzyme-protein ratio used?
Usually the enzyme to protein ratio is 1 to 50.
All right, Rob, we're back to you. Someone wants to know, after reconstitution, how long can IgG0 and FabRICATOR be stored at minus 20 degrees centigrade?
Going based on experience and certainly against the recommended storage conditions for the enzyme, we currently would reconstitute the enzymes and aliquot them out and store them at minus 70, and we can store that for greater than 12 months.
All right, Frederick, we have more time and we have some more questions coming in for you. Are you developing enzymes capable of preparing F or Fab prime 2 fragments from rat IgG1 and mouse IgG1?
We are, of course, looking into this. We have nothing that is really close to being launched at the moment for performing that type of tasks, but this is something that we are currently working on.
Okay. Hey, another quick question. Specificity, how does this work on IgG4s?
Yeah, it works excellent. So you will have the same specificity on IgG4 as you will have on IgG1. I think there's actually a paper published on that on using the FabRICATOR enzyme on IgG4s. I just don't remember it at the top of my head, but I could probably get back to that in a minute.
Okay, well, you know what? Actually, our time is up. So I'm going to thank our listeners for listening in, and thanks for the great questions, and thanks to our presenters as well for presenting today and answering those questions. This webinar will be archived for 6 months on our website, www.genengnews.com. If you missed parts of it, you can watch it again, or you can recommend it to friends and colleagues. And I also want to thank our sponsor, Genovis, for making this event possible.